In this video, we discuss the 6 hallmarks of cancer as originally set out by Hannah and Weinberg, one of the most influential and most cited papers in the last 20 or 30 years. By reading and tens of thousands of papers on cancer, they were able to identify 6 biological processes that have to go wrong in nearly every cell to enable a cancer to form. That 6 processes are required is testimony to the cell's safeguards against cancerous growth. Many of these systems can fail, 4 of them, and you'll still not have a cancer. But eventually, with enough mutations, cells can acquire enough abilities to overcome all the cell's normal safeguards against unintended growth. We'll see that the different processes are self-sufficiency and growth signals, of which RAS is an example, insensitivity to anti-growth signals, the inactivation of, say, retinoblastoma protein, the ability to evade apoptosis when it's called for, limitless replicative potential, for instance, turning on telomerase when it's not appropriate so that telomeres don't shorten with each cell division, the ability to sustain angiogenesis to provide the nutrients to support the growing tumors, and to be able to invade tissues and metastasize. Metastasis involves cells breaking free into the bloodstream, so that means inactivating cadherin, the proteins that mediate cell-cell contacts. Anyway, we'll go through these one at a time and discuss them in a little greater detail. But what's implicit in this formulation is that there are certain mutational pathways that will eventually lead to cancer. In essence, you have to generate mutations in genes that participate in all 6 of the biological processes for a cancer to occur. Thus, when we examine, for instance, the cell line here, which accumulates mutations, when it has this one mutation that allows the cell to evade apoptosis, once you acquire a second mutation here, that's not really adding to the metastatic or cancer potential. You've already fulfilled that capability with the first mutation, so in a way the second mutation is redundant. You see in these pathways a general bias towards self-sufficiency and growth and insensitivity to anti-growth at the beginning
of the mutational profile,
and limitless replicative potential and invasion and metastasis occurring in the second part. So those would be later mutations. These mutations providing a proliferative advantage, these mutations supporting proliferated mass. So taking these each in turn, first discussion of self-sufficiency of growth signals. Going to try to always list a few genes that you've heard of before that contribute to this particular biological process. And in terms of self-sufficiency and growth signals, probably the one we've talked about most is RAS, and how the RAS, RAF, MAPK cascade is mutated in 25% of the tumors, providing signals to the nucleus that cells should grow, even if ligands aren't bound. HER2 is one of the receptors that is active in breast cancer, and EGFR is the tyrosine kinase receptor most active in lung cancers, where both of these receptors might fire or produce downstream signals whether or not a ligand is bound. And here we see a cell diagram from the Hannah and Weinberg paper, and we'll just highlight here again mutations in the RAS and the MAPK pathway, eventually going to implicate changes in gene expression that promote cell growth even without ligands being bound. Mutations in the receptor tyrosine kinases themselves can also trigger these pathways. And here is ABL, the tyrosine kinase that is expressed at high levels and mutated in the Philadelphia chromosome. In terms of insensitivity to anti-growth signals, insensitivity to those signals that tell the cell to stay put and not divide. It's the inactivation of the retinoblastoma protein, which when working normally prevents cell cycling. TGF-beta, which will directly stimulate the retinoblastoma protein, also will, when functioning normally, block cyclin CDK complexes. And we'll also talk about c-MYC a little bit. This is a protein which keeps cells in a pluripotent state. When c-MYC is normally turned off, that's a clue for a cell to differentiate and go into terminal differentiation to be a mature adult cell. We'll see in stem cells when we want to make the induced pluripotent stem cells, one of the 4 genes that gets turned on is MYC, and that will take a mature differentiated cell, de-differentiate it into a stem-like cell. So here we focus on retinoblastoma, again, which will inactivate the transcription factors which promote cell cycling. TGF-beta is a ligand for the TGF-beta receptor, which activates a pathway that will influence retinoblastoma activity. And here we have MYC, which is actually influenced by the MAPK pathway. Again, this is a heterodimeric transcription factor that will cause dedifferentiation through expression of a wide variety of genes involved with a more stem-like state. Now, normal cells will undergo apoptosis when DNA mutations and damage accumulates. Cancer cells learn to evade apoptosis despite a lot of DNA mutation and DNA problems. Far and away, the most statistically important mutation is that done to p53. p53, when working properly, will cause cell cycle arrest or apoptosis. When p53 is mutated, neither cell cycle arrest or apoptosis will form, allowing the cell to continue to cycle, to continue to accumulate more mutations. Now, normally, BCL2 prevents apoptosis. When it's called for, when DNA damage accumulates, a normal BCL2 will stop being present, stop being active, therefore allowing apoptosis. However, mutations can occur in BCL2 such that it will prevent apoptosis even under conditions where it should allow it. And here we see p53, the integrator of DNA damage sensing. We see its direct effects on gene expression and also on inhibiting pathways of cyclins and CDK that contribute to cell proliferation and cell cycle arrest. And here we can also see its influence in apoptosis, the Bax protein, which can contribute to pore formation, which is blocked by normal BCL2 for healthy cells, and then this pathway funnels into apoptosis, which is the desired result in the context of lots of mutations. With cells dividing beyond the Hayflick limit, we have to have limitless replicative potential so that we have to elongate telomeres, as is done in early embryonic development or in pluripotent stem cells. The enzyme responsible for that is telomerase, and in 85 to 90% of tumors, telomerase is upregulated when it shouldn't be, therefore keeping chromosome ends long, allowing them to divide essentially an infinite number of times. P16 protein inhibits the cell cycle when functioning normally, and it also regulates the expression levels of telomerase. And there's not a great deal here, but you see P16 being a regulator of the RB protein and in some manner influencing a pathway that is going to increase the expression of telomerase, which is not here on this diagram, leading to elongated chromosomes ends. For tissue invasion and metastasis, we have to have proteins like proteases that are going to cleave existing tissue and create space for a tumor to grow. We're going to have to break through the basement membrane, and we're going to, in order to achieve metastasis, going to have to break through the requirement that cells are attached to each other. So here we'll see that proteases, collagenases, integrins, and cadherins are all effective in these biological processes. And here we have a diagram. Here in yellow is the basement membrane sitting underneath the epithelial sheet, which again is the layer of cells that line things like the lungs and digestive tracts. 85% of all tumors originate in epithelial cells. Here we see a mutation that's going to cause a population of cells to grow more quickly. A third mutation increases the rate of division and growth even more compared to the cells that are surrounding here. And then we'll start to refer to this as a clonal selection, meaning that one original cell has given rise to a large number. And eventually we have what causes a tumor to be called invasive, meaning it's broken through this basement membrane. It's going into the fibroblast and stroma layers underneath. Now it's going to have access to the blood tissue and the lymph tissue. It's going to have avenues for spreading, and this carries a bad prognosis in almost any tumor setting. In breast or prostate tissue, you have ducts, again lined by epithelial cells surrounded by basement membranes. Carcinoma in situ is a proliferation of epithelial cells filling in the lumen of these ducts. I think the terms benign and malignant are not so satisfying, but localized tumors considered benign most of the time. And in malignant or invasive tumors, again, it may be filling up the lumen, but it is also broken through the basement membrane again into surrounding tissue. And here we see how once it's broken through the basement membrane, it has access to the bloodstream. Via mutations again in the cadherin proteins, now cells can survive on their own, not connected to each other. They'll go into the bloodstream, and eventually they're going to go in the inner linings. They're going to attach to the endothelial wall, and a few cells will be able to make it through the endothelium into a location and eventually colonize the formation of a metastatic lesion. Now this is a very rare event. Cells have to gain a lot of new abilities to be able to break free, to be able to fit into through the endothelial layer, and to be ready to colonize a different tissue type or milieu. But unfortunately, once a tumor is palpable, even though it's a rare event, we're shedding lots of cancerous cells every day. Some estimates have put a minimum of 10 million cells a day sloughing off into the bloodstream once a tumor is large enough to be recognized. If only 1 out of every 10 million cells is able to establish a foothold in different tissue, that's still 1 metastatic growth per day. Cells have to acquire a lot of other abilities for metastasis, like for instance deformability to get through the endothelial cell, mobility to be able to move once it's in tissue. And so a wide variety of genes will affect this biological process. And here, just so we don't always look at cartoons, here's a normal epithelial layer. Here we also see keratin sheet that have formed. We see an excessive growth in one area. This is an intraepithelial layer, neoplasia or tumor. Here we see a higher grade just because the proliferation is much more apparent. And finally, breaking through the basement membrane, which is normally right around here. And now we have an invasive cancer rather than localized ones. So now that we have the accumulation of tissue, we need to have angiogenesis because the cancer needs nutrient supply in order to survive. VEGF, vascular endothelial growth factor, is by far the best known gene involved in this pathway. Its role was demonstrated by the seminal work of Judah Folkman, who rationalized that inhibiting cancers can be difficult because they have ways of mutating around, but if you were able to inhibit the formation of quote-unquote normal tissue, normal blood vessels, then you could essentially starve the cancer and treat it that way with attacking cells that are less likely to be able to mutate and respond to the drug. Other genes that influence angiogenic activity are the Sark-like tyrosine kinases, and we'll see some examples of those. Here you can see Sark, which when mutated leads to increased angiogenesis. It's in the way it's influenced by growth factors such as like the VEGF growth factor. And what's happening here in angiogenesis is then, as long as cancer cells are localized, they're not going to be in communication with the cellular tissue underneath. Once it breaks through, molecules which are released are going to be released into the stroma. This is going to form a gradient that's going to stimulate growth of blood vessels to help feed the tumor. And now not only do tumors have better nutrients or environment to grow even faster, this also gives them an avenue to break off into and metastasize. So while blocking angiogenesis remains a very attractive modality of treatment, unfortunately the clinical trials have not worked out quite so well. Nevertheless, VEGF inhibitors like Avastin are approved for use in several different cancers, only that the effects have been a little more modest than were hoped. So now that you know about the 6 categories or biological processes that a cancer cell has to acquire the ability to take care of, whenever you look at a cell, you can identify the mutations that are present. You can start to make predictions about what kind of mutations would cause the neoplastic transformation of those cells. And looking at a set of possible mutations that might cause cancer, you can immediately pick this one as the key set of mutations that's going to cause invasive cancer, insensitivity to anti-growth signals, and metastasis and tumor invasion. When combined with the existing mutation complement, will give a cell the capability of doing all the processes cancer cells need to achieve if they're going to grow in an unregulated fashion. Okay, thank you very much for listening to this